Okay, so I've been having a bit of technical difficulties today, a lot of technical difficulties. But we're gonna get through this. We're almost at the end, even though it feels like it's never ending. I'm sure you guys feel that way as well. So we are on to other molecular techniques, moving on to DNA sequencing. So this has completely revolutionized, another thing that has revolutionized genetics. It's how we basically characterize our DNA, right? We know everything about our DNA now. We can isolate it, analyze it, look at its sequence, find any non-coding part or coding part, look at the introns, because now we realize that they're consensus sequences that distinguish introns apart from actual expressive exons. We can look at regulatory sequences. It basically helps us completely categorize and characterize and organize even the genome. So it's extremely important. We'll start off with talking about the first automated technique, which was the dideoxy or chain termination sequence that was pretty much invented by Frederick Sanger. He received a Nobel Prize for this. We still use this technique for really small fragments of DNA, so small scale sequencing. But for the most part, sequencing has really come a long way. We've proceeded on to moving on to things like next generation sequencing or even third generation sequencing, which have made not just sequencing itself more efficient, but also, amazingly enough, even less expensive. This is one of the main reasons why the Human Genome Project didn't take as long as projected, right? It didn't take as long, and it wasn't even nearly as expected as they had previously thought it was going to be. So they actually ended even earlier because these sequencing methods and mechanisms got better and better and better, so much so that they can sequence millions of base pairs within a day, even probably in an hour's time. While before, a million, like 1,000 base pairs, it would require a few days to do. So let's talk about the dideoxy or chain termination sequencing method. This sequencing method, essentially, the key component of the sequencing method is the dideoxynucleotide, okay? So the dideoxynucleotide, as I'm going to show you here, you're going to see it drawn a couple of times because amazingly, I've done this video twice before. I've told you I've had really bad technical issues. So dideoxynucleotide, basically is a nucleotide that has one less oxygen. So it's not an actual mistake, it's not a typo, it's dideoxy because 2 oxygens now are missing. Now your typical deoxyribonucleic acid, which is a nucleotide that you have in your DNA, looks like so. You have a 3' OH and then your 2' is missing an OH and has a simple hydrogen, right? And this is what RNA looks like. So, and this is what one of the things that distinguishes RNA from DNA. But in a dideoxynucleotide, the 3' OH does not have an O on it. It's actually just a hydrogen. And hopefully you guys have heard me say it over and over again. But by having just a hydrogen is going at the 3' end, this is going to stop DNA polymerase from working. So DNA polymerase requires a free 3' OH end in order to add its nucleotides to it. If that 3' end is now only in hydrogen and not in a hydroxyl group, an OH group, DNA synthesis terminates, okay? So, and this is basically, these dideoxynucleotides were used to carry out sequencing. How were they used, you may ask? So, they did an experiment where they cloned the DNA and they've separated all these clones into 4 tubes. So each tube had the clones of the DNA that they wanted to sequence. In one tube, they added the DNA polymerase and all the normal deoxy triphosphates. If you guys remember, NTPs or nucleotide triphosphates are going to be the raw material that the DNA polymerase just adds on while reading the template DNA to synthesize the new DNA strand. And then in one tube, they added all these things and they added a small amount of the dideoxy adenine triphosphate. Okay, so a small amount of the dideoxy adenine. Then they did in the other tube, they added all these ingredients and they added a small amount of the dideoxy thymine. Then the other tube, they got all the ingredients, added the dideoxy guanine, and then the final tube, all the ingredients, all the normal nucleotides, plus the dideoxy cytosine. Okay, so this means that occasionally, randomly, the DNA polymerase, because the dideoxy ATP, even though it's missing an OH, it will still be added. Okay, randomly, the DNA polymerase would add, excuse me, would add this dideoxy ATP. would add the adenine every time it you know, it was necessary. And as soon as it added the dideoxy ATP, it would terminate the synthesis of that particular fragment. So for example, here we have this one clone, and then they inserted the adenine in the first time the adenine was supposed to be inserted. And so we had 3 bases and an adenine right here, okay? The next clone, randomly, they incorporate the first adenine that the DNA polymerase incorporated was a normal adenine. And then in the fifth, like the second adenine, the 5th base pair, it randomly incorporated the dideoxy adenine. And it continued to do so until we got all these fragments, and we knew that these fragments of different length, right, of different length, and we knew that at the end of these fragments, we had an adenine, okay? So because we knew that that was what we had in that tube. We did the same with the second tube, where we had the thymine, the third tube, where we had the guanine, and the fourth tube, where we had the dideoxycytosine. And so basically in this way, they then separated it on a gel, and they, like these fragments on a gel that was so specific that it was able to separate even 1 base pair in length in a fragment. So, and by looking at which base pair came first and which which fragment came first and which one came second, and etc., they were able to basically put them together. So if you see here, I have GATC, okay, GATC. So here, the first band is a cytosine. Okay, so that's my first base pair. The second one is fragment is probably just 1 extra base pair away from that cytosine fragment. So that means the last one is adenine here. So that means in this fragment, we have a C and an A. Okay, then we have a G, then we have another A, then another C, then an A, a G, a C, a T, a G, a T. Do you see how that goes? So each fragment increased by 1 base pair, and that 1 extra base pair was the dideoxynucleotide. And whatever that dideoxynucleotide was A, T, C, or G, it basically showed which base was in that position. So this was a Sanger method. It's actually pretty like phenomenal, if you ask me. They then made the Sanger method a little bit better by basically tagging each dideoxynucleotide with a fluorescent tag. This fluorescent tag could be blue, like different colors. Each one had a different color, and they all put it in 1 single tube. So this made things a little bit quicker, a little bit faster. And it was essentially the same principle as the Sanger method, but now everything was in the same tube and everything was tagged. Okay, so they have fluorochromes, everything was tagged. So this was basically how sequencing started off in the first place. And then in the last 20 years, next generation sequencing techniques have been developed that are much faster. DNA fragments are amplified, so we have a lot of the same identical fragments. And then 1 strand of each fragment is immobilized, and the complementary strand is synthesized 1 nucleotide at a time. And then we have electronic monitors, which are used to identify in real time which of the 4 nucleotides is added. And one of these nucleotides all have different fluorochromes, so they're all tagged with different fluorescent tags. And so it's basically, you see sequencing by synthesis, right? And so each different, you can see here, this is known as a chromatograph. I'm gonna try making this a little bit bigger, a chromatograph. So each of them it's just basically the fluorochrome that comes up, right? And by reading it, it's just electronically and seeing which fluorochrome fluoresced at that specific time when the synthesis of 1 base pair happened. You can figure out which nucleotide was added. And in fact, actually, when you send out your sequence, you usually always get a chromatograph back right here. Really clear peaks, really tall peaks like this means you have a really clear, good sequence. When something looks more along the lines of these little peaks, if you can't see them, they're just kind of like weird little lines. That means that the sequencing wasn't very accurate. It probably has to do with DNA, your DNA sample not being that great. So that's just something about sequencing. So this is an example of high throughput technology, because at this point, hundreds of thousands of fragments, about 300 nucleotides long, can be sequenced in parallel. The method of choice this is a method of choice when you have lots of DNA samples, like numerous fragments of DNA and etc. Right? Okay. So this is like, yeah. So the next generation sequence is often complemented or even replaced at times by 3rd generation sequence. Here, like each technology is basically faster and less expensive than the previous one. So it's the same new methods of DNA. It's like there's some new stuff that happens with third generation sequencing. Here, the DNA does not have to be fragmented. So you can make like a long molecule of DNA and sequence it all on its own. The technology developed in which is basically a single strand of DNA is moved through a nanopore, essentially, so a small pore in a membrane, and then you identify the bases one by one by the distinct way, like each interrupts a specific kind of electronic or of an electrical signal or an electrical current. Okay. So the idea behind this is that each type of nucleotide intercepts the electrical current for a slightly different length of time. So this is basically just improved DNA sequencing techniques, and they've transformed the way in which we can explore fundamental biological questions about life. Sorry. Yeah. So the next part is creating knockouts and transgenesis. This is a really big, huge molecular methods that we use to study genes and their function. So knockout means you take away the gene, you knock away the gene from the organism, and then transgenesis is you put the gene in another organism and see how it works. So they're both kind of similar to each other, but they're just ways to study gene function. So gene targeting and knockout animal models. So gene targeting, the concept is to manipulate specific alleles, loci, or base sequences and learn its function on the gene of interest. So this manipulates a specific gene in the genome. The gene knockout concept is basically eliminating the gene in an organism and seeing then what happens. What's the phenotype that arises if we shut down a gene in an organism? And by seeing what occurs, or what doesn't occur, we can figure out and learn a little bit more about the gene function. So knockout, like the first knockouts happened in mice, and this really revolutionized research. Now we can do it in pretty much a lot of different organisms, like zebrafish, fruit flies. Y'all know about fruit flies. Lots of different kinds of organisms where we do knockouts and we just get rid of the genes. This is a very intense and long process. It's very time consuming and even quite expensive, as we'll see soon. There's a lot of like mating, like you have to mate your different heterozygous knockouts. Like it will make sense in just a second, but obtaining a fully knocked out mice does take a while. But then once you've established one knockout mouse, you have basically a generation of those knockout mice. So they are worth it in the end. So how do we create knockout mice? So to make knockout mice, sorry. So we need to construct a target vector. So you have to have a vector with your target gene in it, right? And you want to make sure that the target gene that you have inside your target vector is mutated, because essentially what we're going to do is we're going to add the target vector to the mouse cells, and then this target vector, the gene that it carries, it's going to make the normal gene in the mouse switch places with the mutated gene that we just brought in. And so basically this means that the mouse no longer has a normal gene and now has the mutated gene. So let's see if I can say that again. So let's actually read it. So the target vector has a mutated copy of your gene of interest, okay? And a selectable marker, because we have to make sure then that the cells that obtain this target vector like that, actually we have to make sure that we need a way to certify if the cells obtain the target vector or not. So the target vector has your gene of interest in it, and your gene of interest, because you want to knock it out, is mutated, so non-functional, okay? So when your target vector is inserted into your cells, and these cells are usually embryonic stem cells, okay? It's inserted via electroporation or direct injection. So the direct injection kind of looks like this. Let me tell you guys, it's very difficult. You're under a microscope injecting them, it's insane. So when you're putting in your target vector through electroporation or direct injection in your embryonic stem cells, once, like say it works, once it goes inside the target vector, once the target vector goes inside the stem cells, at that point through homologous recombination, you have the gene of the mouse, which is nice and intact, and then your target vector coming in with the bad gene, okay? the gene and the bad gene, the mutated gene, which are very similar to each other, quite homologous, except for that couple of mutations that you've added in your target gene of interest, right? They come in, and then through homologous recombination, the good gene comes out and the bad gene on the target vector goes in, okay? And then the genes that were out, like the target vector, the gene that came out will be degraded. But essentially what occurs is that now the gene that you brought in, the mutated gene that was brought into the target vector, is now found within the chromosome of the mouse, right? Or the stem cells of the mouse. So randomly, some embryonic stem cells can take up the target vector, the targeting vector. This is really difficult, guys. It's very hard to get a cell to pick up a vector. Trust me, it takes a really long time. And it takes a lot of luck. So if some of them do, the few that do, an endogenous enzyme recombinase catalyzes homologous recombination, and then the target disrupted gene will replace the gene you are knocking out on 1 or 2 chromosomes of the mouse. Okay, so once you have this happening, then you have to actually select for the embryonic stem cells that took up the targeting vector and the ones that did not take the targeting vector, because there's no way you can look at them and see which one has it and which one doesn't under the microscope. So remember how our targeting vector also has a selectable marker. So this is how we're going to select our recombinant embryonic stem cells. So our recombinant embryonic stem cells are selected depending on the selectable marker, and they're injected into the mouse embryo at the blastocyst stage. I thought I was going to explain more. So they're selected because the selectable markers can be neomycin resistant, right? They can have something like GFP, a green fluorescent protein attached to them. And so we can actually see the embryonic stem cells that are resistant to neomycin. So if they are, like we expose the embryonic stem cells to neomycin, the ones that don't have the neomycin resistant gene that came along with the target vector are gonna die. And the ones that do have it, because they also have the target vector and the mutated gene of interest, are going to live. And so they're called the recombinant embryonic stem cells. We can also use GFP as well, or LAC-Z, or whatever kind of really like a nice, easily used selectable marker gene. Okay, so once we select our recombinant embryonic stem cells, they're then injected into a mouse embryo at the blastocyst stage, where they will then be incorporated into a surrogate mother mouse. So we put them in the blastocyst stage. I don't know if you can see. The blastocyst stage is just when the cells are starting to develop. So we have 4 or 6 cells or 8 cells like just coming about from that first zygote, from that first fertilized egg. We inject it in there. And then once that happens, we take it and we put it into another mouse, a mother mouse, and she will be pseudo-pregnant. So she's not pregnant with our actual child. She's pregnant with this new blastocyst that you made, that has the knockout gene. So the result is that once the pseudo mom goes through labor, the result is a chimera. So chimeras have some cells that come from the knockout stem cells and other cells that are just your normal stem cells of the donor blastocyst. So if you see here, I believe this is the blastocyst. I think this is what this picture is saying. So some actually had obtained the recombinant embryonic stem cells, others didn't, and so we kind of have a chimera. And you can see the mice here are kind of half-half. Chimeras mean half something else. So some of them have the knockout stem cells, other ones don't. That means some of their cells have the knockout gene, other guys don't. So in order to get a mouse that has basically all knockout, we mate the chimeras together. And this gives us a homozygous for the target gene. Okay, so this target gene now, we've disrupted it, it's mutated, so it's non-functional. And we now have our knockout mouse, and our mouse can live forever. So depending on what you're actually even studying, right, depending on what your gene does, your knockout mouse can be conditional, meaning you can control when the knockout turns on. Because a lot of times some genes are important for embryonic development, right? And if we knock out the gene as soon as like the embryo is forming, guess what? The embryo is going to die. Your knockout mouse is going to die because you knocked out a really important gene for embryonic development. So what we do for these kind of genes or other genes where we want to learn kind of like more of their regulation, we make it conditional. So we allow the knockout gene to to stay basically hidden, to not be expressed for a while, to express normally actually for a while. And then once it's past the crucial development stage, or once it's past a specific time and we want to learn more about it, we turn it on and then we see what happens. And so by seeing basically the phenotype that comes out once we knock out a gene, we can figure out what happens, right? It's a little bit drastic, I have to say. I don't like making knockout mice. Anyways. So then we have transgenic animals. So this is the opposite of knockout animals. We have knock-in animals. Knock-in animals are basically when we purposely put in a gene for them to express. So they are expressing or overexpressing a particular gene of interest. So it's transgenic, because usually it's a gene from not even just another species, right? It can even be from the same kind of species, but you're still putting it into another organism, so it's still transgenic. This is a lot easier than knockout because the gene simply needs to be incorporated into the host genome somewhere, preferentially in a non-coding region, and not at a particular locus, right? So that the targeting gene, you have to make sure the targeting, like the knockout, the targeting vector needs to go to a specific area and knock out the gene at a specific location, specific locus. Here, the new gene can be inserted wherever, whenever. So the vector with the transgene undergoes, again, homologous recombination into the host genome. There's a variety of different ways we could do this. Transposons actually are used a lot. If you remember transposons, jumping things, those things that allow jumping stuff. We can put a vector with transposons on each side of the gene of interest, and then once it goes inside the organism, the transposons, we can activate it and the transposons jump, and they make the jump into the actual genomic DNA, the actual chromosomes of the organism. There's a variety of different ways to do it. So the vector with the transgene can also be put in the embryonic stem cells and injected into embryos. Again, very invasive process most of the time. So much like the embryonic injections that we saw before with the knockout mice, it's just a lot of times the needle is so invasive that the cells don't develop properly, and you get kind of like a little monster. Anyways, yeah, they allow for studies of the effects on the appearance and function of genes. So you can even decide to overexpress certain genes and see, that's beneficial to understand how cancer works, the expression of human genes or genes from other species, or the expression of mutant genes as well, and see what could possibly occur if these are mutated, if these genes are mutated, right? So very, very valuable technique, extremely valuable technique. Transgenesis is a big deal. It's done? Oh my gosh, it's done. I thought we were going to talk about gene editing. We're going to talk about gene editing. So gene editing is another really big molecular technique that has become a lot more prevalent, right? So I'm actually amazed that we're not talking about CRISPR here. But anyway, so CRISPR is one of them, right? So gene editing, that's more, has like a medical application to it, because you guys know better than me, right? So there's a lot of genetic disorders basically stem from one single nucleotide that is changed. Okay, one single nucleotide that's changed. And for the longest time, scientists were trying to figure out how we could change that one single nucleotide in a person that suffers from a really bad genetic disorder, right? And how we can basically change their life around. So there was a lot of different experiments with retroviruses. So basically injecting or infecting the person with a modified retrovirus. And you guys know that what retroviruses do, or what viruses do, they infect you, they inject their genome in your cells, right? So a particular study in France did this with a few kids that had a particular immune deficiency disorder, like 10 things. I think this was in the 2000s, like early 2000. And they injected them with this retrovirus that had the correct gene in it, right? And they injected the little kids when they were young in their bone marrow cells, because that's basically what the ones that would reproduce, and where the majority of the cells could then have the new gene. And so if the majority of the cells have the new gene, they at least could make the correct enzyme and basically counterbalance the incorrect enzyme that was made, or the lack of the enzyme that was made in a particular disorder. So they gave the kids these retroviruses, and with the new gene, right? Then the retroviruses injected this new gene and essentially the rest of their genome, their artificial genome, inside of the kids. 9 out of 10 had a really good, 9 out of 10 were able to get better, but then down the line, 3 of them developed leukemia. And that's because probably the retrovirus inserted itself in a place in the genome near some cycle, near some gene that was important for the cell cycle, because there's no way of targeting where the retrovirus gene is actually inserted when it goes inside your genome. So there was a couple of studies with those, that for gene editing. And then around 2012, I believe, they discovered a way to use the Cas9 system. Now, the CRISPR-Cas9 system is something that is already that happens naturally in bacteria. It's actually one of the ways that it's one of their defense mechanisms towards getting rid of viral attacks, right? Because viral attacks, again, they insert their genomes inside the bacteria, etc., or humans. But these bacteriophages, this is how the bacteria would protect themselves from bacteriophages. And so they realized that the Cas9 was very specific. Cas9 is the actual protein involved. So this protein was guided by a guide RNA. I'm going to open up my other slides from another class, sorry. I think this is important for you guys to know. The guide RNA that was complementary to, the actual sequence of the virus, and then it would bring about Cas9 to the viral genome, okay? And then the Cas9 would basically cut everything up. It's an endonuclease. It cuts the, I opened up the wrong chapter, sorry. It cuts the actual DNA. And so they saw that they could actually make the Cas9 go ahead and cut, just like a scalpel, like a molecular scissor. They could make it go and cut in specific areas by changing the guide RNA and making it so that it was complementary to whatever target gene they were looking at. Okay, here we go. Sorry for taking so long. So here, so it's most widely used tool for altering genomes in vivo. It allows direct modification of genomic DNA in any cell into which DNA can be introduced and expressed. Projects are varied. They're really cool. Some of them have been involved in removing allergens from peanuts. That's pretty neat. And then others, which are even not still in clinical trial, basically are to cure genetic diseases in humans. So these have been more proposed. A lot of it's been kind of put on hold just because of the ethics behind it, and because of the fact that we don't know if there's any off-target processes, off-target working. It's Cas9 could accidentally go and cut some other kind of gene and inhibit some other kind of gene that's very similar to the gene it was targeting. We don't know. So there have been a couple of human studies. There was one in China where they took basically non-viable embryos. They took non-viable embryos and they wanted to, there was a specific gene that was necessary for a disease called thalassemia or something like that. It's a blood disorder. And anyways, it's a blood disorder. And they were able to stop the expression of that particular gene with the Cas9, but it caused the expression of other genes around it. And so because of that, it was okay, there's a lot of off-target processes. And so a lot of this has to be studied a lot more before we can use it on humans. But then this is illegal in some countries, right? In other countries like China, it's still going, I think. Anyways, so how does it work? We have genome editing. So Cas9 is a nuclease. It can cleave both DNA strands, and then it's followed by homologous recombination. So we can guide the Cas9 with the guide RNA to our target gene. And then with this guide RNA and Cas9, we also include the donor DNA that has the correct gene sequence, right? And then as soon as we have that cut, the homologous recombination comes in. Homologous recombination, which is already present in your body, okay? It comes in and it switches the donor DNA with the bad mutated DNA. Boom, it switches it over. And so now the person has the good DNA and the non-mutated DNA, the donor part that you put in. And essentially, that's basically how it works. That's basically how it works. So this could have a myriad of different kinds of applications, but there's a lot of different like opinions on this. I believe we still need to research it a lot more just to try and prevent those off-target sites, because we don't really know as much as we should. There's a really good Netflix, I already told you guys about this, is a Netflix documentary. You guys actually told me about it in the first place, called Unnatural Selection, that I highly suggest you guys look at and watch to basically figure out what's happening. Okay, so that's a little bit on gene editing. Very important. Gene therapy is a big thing, and I feel like it's going to really blow up in the upcoming years. That's it for chapter 17, guys. That's it. So end of chapter 17, end of genetics lectures. You guys did it. Push through just a little bit more. You're almost done with the semester. I'm proud of you guys.